Hi, welcome back to my channel. My name is Jeffrey. In this video, I'm going to discuss how to study for medical school. Okay, so this is just a document that I imported from Notion. And essentially, what is in this document, it just consists of all the lecture objectives. And this is just from lecture one, right? And as we scroll through this document, you can see that I'm still scrolling here, and it's not like I'm scrolling really slowly, right? So at the end of this document here, you can see that it's 18 pages long. Now, you might be thinking, holy crap, that's actually so much material. How do you go through all of it? And this is just one lecture, and every day we go through two lectures, right? So here, I'm going to just tell you guys, there are a lot of things that you don't have to know, and how you know that you don't have to know that material is just by using your brain and thinking about it, right? So I listed out the lecture objectives here. Let's just go through them, right? So list the domains of cellular life: bacteria, archaea, eukaryota. Okay, yeah. How are they going to test us over this? Well, the most likely thing is just like eukaryota, it has membrane-bound organelles, and the other two don't. Right? That's the only thing that they can possibly test you. And this is actually so low yield that I would say just not even just answer this question in your head, right? List the domains of cellular life. Well, it's common sense. You've learned this since high school, since middle school, right? You learned bacteria, you learned about archaea, you learned about eukaryotes, and you learned about what makes eukaryotes different from bacteria, right? It's just that they have membrane-bound organelles, and that's literally it. So don't even waste your time writing out notes for this because that's a waste of your time. So the second part right here, it says ID, list and describe the structure of the following: nucleus, and then down you have like nucleolus, your ribosomes, so on and so forth. So right here, I wrote down 10% of the cell volume. It contains DNA. Well, no shit. Everybody knows the nucleus contains DNA. Why did I write this? I don't know. It was a waste of time. Do I have to know that it's 10% of the cell's volume? No, it's not important, right? Just know that the nucleus is small compared to the rest of the cell. And then here you can actually just see this, that's the actual nucleus, and then here in the center is going to be the nucleolus, which is the dense part, right? So nucleolus, I said the site of ribosome production and assembly. This might be actually important, right? Because we know that for proteins, they get assembled in the rough ER. They get assembled via ribosomes, right? So the fact that this ribosome production assembly is in the nucleolus and not the rough ER, that's very different. That's something that they can test you on, and something that's clinically significant, right? How might they test me on this, and how might me as a doctor go about treating disorders, right? So ribosomes, if they're defective, then you're unable to produce protein, right? So it's very important. So there are three zones, and it has the fibrillar center, the fibrillar material, and the granular material. Do I have to know this information? It contains rRNA genes. This transcribes rRNA genes, and then you have your ribosome assembly here. Well, it's very important, right? So your fibrillar center obviously contains the DNA, the genes for rRNA synthesis. And then in your fibrillar material, that's where you actually get transcription of those genes, right? Granular material, that's where you assemble them. So three majorly different functions. And so this slide right here, this histology slide, is going to be very important, right? It could be something that they're going to ask you to identify. They're probably just going to blur out these areas and then just give you arrows and say identify, right? So you just have to know that the granular material is going to be very dark. You have to know that your fibrillar material is going to be darker than your fibrillar center, and your fibrillar center is very pale, right? So that's something that you have to drill into your brain, and that takes time, and that takes effort. But just remember that the time and effort that you're going to spend on that is all the time that you save from not writing this bullshit down, stuff that you don't have to know, right? So then here we come down to nuclear envelope, the inner nuclear membrane, outer nuclear membrane. Like, do I have to know that the inner nuclear membrane faces the nucleoplasm? It contains nuclear lamina, structural support for the nuclear envelope. Well, if it's on the inner side of the nuclear membrane, it's common sense that it's going to be touching your nucleus and like be closer to the nucleus, right? And then for your outer nuclear membrane, it's continuous with the rough ER. That makes sense because the rough ER is right outside of the nucleus. So then for your nuclear envelope, which is literally right outside of your nucleus, then your outer nuclear membrane is going to be closer to the rough ER. It's going to be continuous with it, right? So that's just maybe things that are mental notes that you have to make. Maybe these are not things that you have to use time to write down, and it's something that you can reason through, right? Using your knowledge of biology, using your prior knowledge from MCAT, from all of your different biology classes, microbiology, you can reasonably assume that the inner nuclear membrane is going to be next to the nucleus. And then the outer nuclear membrane, well, that's going to be close to the rough ER because it's on the outer side of the nucleus. And that side is with the rough ER because the rough ER has all of the ribosomes to make your protein. And you want to make that close to the nucleus because remember the central dogma, right? So the central dogma or central dogma says that you have your DNA to your RNA to your protein, right? So you don't want the RNA to degrade before you make your protein, so that makes sense, right? Just spatially. So just knowing these words, making the connections with these words, rather than writing down a bunch of notes and hoping that you can brutally memorize all of these different words, I think is more important and easier, right? So having this picture here, wow, look, the nuclear lamina is on the outside of the nucleus. You have your nuclear pores here, right? So nuclear pore, it says, I wrote, spans the nuclear envelope 80 to 100 nanometers. It's made of nucleoporins. There's 3,000 to 4,000 nuclear pore centers in a nuclear envelope. There's less than 90 nanometer diffusion, and over 90 nanometers has active transport. So what that just basically means is if it's under 90 nanometers, it's just going to diffuse right through, right? Is what's important here? Maybe the nucleoporins are important. Everything else is actually bullshit, right? Like, are they really gonna test me? There's going to be 3,000 to 4,000 channels. You have to over 90 nanometers. It's going to be active transport. Um, Obviously not, right? I was stumbling over nanometers and millimeters for the longest time there. Obviously, you don't have to know this, right? Like I didn't memorize it. It's not important, right? So here is just a histo image of your nucleus, and these are your nucleoporins, right? So this is something that's important, right? This is something that they can test you on. Oh, what is this? What am I showing an electron tomography of, right? Electron microscopy of, right? So here we can go on. It says describe the organizational structure of chromatin. You have your nucleosome. Nucleosome is a structural unit. It has an octamer core of histones. It has two loops of DNA around the core. Do I have to know that it has an octamer core? Probably not. I wouldn't. I mean, it would be nice to know. It'll probably help down the line, but is it really that important? I would just focus on the histones and the DNA part. That's where I bolded, right? So when I went back over this, that's what I bolded because that's what was important. Do I have to know H2B, H3, H4, H2A, H4, H2A? No. Do I have to know histone H1? Well, this is actually a very important protein, but in the context of what you learn, I don't think it's that important. It basically just supports this DNA as it winds around the histones, right? It's not that important. It's nice information to know, but you got to focus on what's important, right? There's no way they're going to test you on this. Is it really that important to understand that it's an octamer core? I don't know, right? Just if you have time, obviously try to memorize all of that information because everything is technically testable, right? You have your two types. You have your euchromatin, heterochromatin. Yeah, I think this is very important because euchromatin, if you take a look at it, it says it's transcriptionally active. Heterochromatin are silenced genes, right? This is very, very clinically significant because a lot of drugs, they deal with your heterochromatin and your euchromatin, silencing genes, certain genes, so that they cannot be expressed, so that you don't have a disease phenotype, or activating certain genes so that you can combat certain diseases, right? So these are very important. And then you have the histo image right here. You can see that there's mitochondria here, the rough ER here. You can see that this is a nucleus. This is probably the nucleolus, right? You have your E labeled for euchromatin, H labeled for heterochromatin. That's definitely something that you have to understand. And if you just know that heterochromatin, since it's wound together so tightly and it can't be accessed by transcriptional material, then you can understand why these areas are silenced, right? Because they're so tightly wound together that you can't even transcribe the genes. For chromosome structure here, we have the centromere, telomere, replication, origin. These are all things that you learned in your intro biology classes in undergrad, in high school, right? You have your centromere right here. You have your replication origin. You can have many of them throughout, right? You have your telomeres on the ends of your chromosomes. So do you have to know that it's a centric heterochromatin? I don't think so. It holds sister chromatids together. That you already knew, but that's probably what I would remember, right? And then telomeres here, they're on the ends. They have telomerase, right? To elongate those telomeres. You have your replication origin. I mean, if you don't know what that is, I don't know what you're doing in medical school, right? You have ribosomes here. You already knew this from your notes before. You have polyribosomes, so multiple ribosomes on a single mRNA. That's probably really easy to remember. I would make a quick note of it, just like I did in here, right? So here I wrote you have two types. You have your membrane-bound ribosomes as well as your free ribosomes, right? So these are just things that you have to just remember, right? So membrane-bound, they're going to be attached to a rough ER, and they have a signal sequence. I um, I wouldn't even know if I would memorize all of that, right? It's just know that any membrane-bound ribosome is going to secrete things outside of the cell, and then you have your lysosomal stuff, right? Just like these things are going to be moving, and if you remember that they're moving, then you can probably remember these three aspects about your membrane-bound ribosomes. So then here you have your signal recognition protein. Do you have to know there's a docking protein, a translocator, a signal peptidase, a signal peptide peptidase, and then your completed protein, your translational arrest here. Like, do you have to know all of that? No, not at all, right? Just remember signal peptide. You have your signal recognition protocol that's going to bind to it, and then that's going to drag it onto the rough ER membrane, right? Don't focus your attention on things that don't matter because what that's too detailed. And there's no drug that you learned so far. There's no clinical correlate on the slides that describes anything to deal with these. So don't waste your time memorizing it. Okay. Um, Moving on to ER, you have your rough ER. Rough ER has ribosomes. Oh, that's why they're rough. No shit. You already knew that, right? Don't waste your time writing out these stupid notes when it doesn't matter, right? Like this is common sense. You shouldn't have to write out any notes on this. You have your initial post-translational modifications, whatever, right? Your smooth ER is going to be synthesis of lipids and detoxification. Is that important? Well, yeah, it's very important, right? Because that's literally the only function of the smooth ER. If you didn't know that already, then I would write down a note. If you knew that already, then I would just memorize it or maybe use Anki or whatever you use to kind of remember these different little aspects of the smooth ER. ER lumen, it's a single continuous space. Is that important? No, it's not important. This is not important. I wasted all this time taking a screenshot, writing all these notes about the lumen of the ER. It's actually useless, right? You don't have to write notes on things that are not clinically significant, on things that are not important. your Golgi here, you have your um, different functions. You already knew that Golgi is for packing, right? So right after the rough ER, draw like a concept map, right? And then right after the rough ER, it's going to go to the Golgi for packaging and modifications, right? Do you have to know that it's well developed in secretory cells? Yeah, sure. Because remember, everything that has a that goes to rough ER is going to be secreted out of the cell, right? So if a cell has a lot of rough ER and it has a lot of Golgi, then you can probably imagine that it's a secretory cell, right? Do you have to know that the plasma cells secrete antibodies and the pancreatic acinar cells? Sure, those are neat things to know, but just think about what they're going to test you on first, right? So this is obviously very important, right? Because your COPE2 vesicles, your COPE1 vesicles, retrograde, anterograde, these are all very important concepts. You have to know which side is going anterograde and which side is going retrograde. And the reason why they go onto this topic is because they want you to learn, look, anterograde, this is a big example of anterograde. This is a big example of retrograde, right? So that's very important. Um, and then you have your constitutive, regulatory, lysosomal. Is that important? Yeah. Just know that constitutive is always active. You don't have to write it down unless you didn't know what constitutive meant. So just like understand how to use your time the most effective way, right? Lysosome, just know that. Do you have to know that the pH is 4.7? No, don't memorize that, right? Like, just know that it's acid hydrolase. Obviously, the pH is going to be lower than 7. Remember, acid is going to be lower than 7, and you'll be fine, right? Here, for just like, obviously, you have to know your histology. So just know, like, oh, that's what a lysosome looks like. Oh, that's kind of what a lysosome looks like, right? So you have these different things about lysosomal digestion. Yeah, that's important because phagocytosis is going to be looking something like that, like a Pac-Man, right? Endocytosis is going to look something different. Um, Penocytosis is going to be like cell drinking, right? So these are things that you have to memorize, sure. So that's why I wrote them down. But just keep in mind that you want to pay special attention to things like this: lysosomal storage disorders, right? They have Tay-Sachs, they have Hurler syndrome, they have Pompe, Gaucher, right? Eye cell disease. Do you have to memorize all of this for your first exam? No, no, you don't. I'm going to be very honest right here. I was very, very nervous because I was like, oh my God, I don't know if I can memorize all of these different things, right? Just understand generally where these things are being affected. So what I did was I watched Sketchy for these, and I had a general understanding that, look, this one's going to be the beta hexosaminidase A, right? Yeah, like you have to understand that Tay-Sachs is going to affect your nervous system. So even if you didn't understand, look, like I didn't know um, the specific enzyme that was affected, at least I remembered the GM2 ganglioside. Or if you see a question that says GM2 ganglioside buildup, then you have to know that it's going to be hexa, right? That's affected. So for these other diseases, it'll be nice if you could remember like phosphotransferase 4M6P formation, but just kind of remember the enzymes that are being affected and what it's going to affect, right? The system that's going to be affected. So all of these things, like, do you have to know E2, E3, E1? Do you have to know this 26S proteasomal complex? It's really not important. It's really not. So for a proteasome, just understand that it degrades proteins and it's ATP dependent. So how it does that is through ubiquitin. Ubiquitin binds to the target protein that you want to degrade, and then once it binds, that's a target for proteasomes to be like, okay, get the fuck out of here. We don't want you, right? You don't have to memorize any of this. Like Like what do you have to know about that? You just have to know that this big protein is going to be chopped up into short amino acids, right? Through this pathway, right? You don't have to know any I would know that there's like E factors, but like what the hell? Like you don't have to know any of that. So just understand that there are things that they're not going to test you on because what they're trying to understand from you is just if you understand the general function of a proteasome. Because when you get into the nitty-gritty details of an actual proteasome, then that's where like it's becoming PhD knowledge. You're devoting all your time into understanding how a proteasome works. When you really, as a doctor, you just have to understand. Look, this is the general enzyme. This is the function of that enzyme, and what can I do? Like, what is clinically significant about it? I don't have to understand every little gritty detail like that. E2, E3 is going to bind to it unless it was affected in an actual pathway from medication, right? A drug target. So all of these different things, there you just have to understand. Like for example, for a peroxisome here, it's going to use hydrogen peroxide, right? And a disease that's associated with it is Zellweger disease, right? Do you really have to know what, like, just all of these different words right here? No, not really. Just know that they degrade toxins and they are important in long chain fatty acid metabolism, right? Just know that it has hydrogen peroxide and how that's different from a lysosome, right? Know what it looks like, and you're good. So this is a very, very long video to tell you guys how to study. I hope that it's been helpful. Just focus on what's clinically significant and broad picture, right? Don't get tied down by every little detail because they're not going to be able to test you on every little detail. If you think about it, lecture 1, cellular organization, they're actually only going to ask you one question maximum on that lecture, right? Maybe two if they're feeling like generous, right? Because like it's not important. Like there are more important lectures that are out there, and there's a ton of lectures that they're going to test you on. And there's only so many questions that they're allowed to give you, right? Because you have 3 blocks. Each block is 50 minutes, and you can only they're only giving you like 40 or 42 questions, right? And there's a ton of lectures. So, you're only going to get 1 or 2 questions on every single lecture. So you got to understand what out of, everything that they can test you out of lecture 1, like what makes the most sense for them to test you on? What are they going to use those valuable few questions to try to ascertain your knowledge? Are they really going to ask you like um like what is archaea? Like no, they're not because they only have one to 3 questions that they can ask you on every lecture. They're not going to waste that 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 question asking you something really dumb, right? Asking you something super super specific like do you 3,000 to 4,000 nuclear pore channel? Like who cares? No one cares, right? Understand what's important. Understand, like maybe they'll ask me about nuclear pores because that's important. Like you gotta like you have to know that there's not a ton of things that can just willy-nilly just go into the nucleus and come out and have like no consequence, or because like that'll be devastating to our cell, right? So understand what is important and focus on that. They're not going to ask you about some translocator or some signal peptidase, right? Because that would be actually evil. That would be so low yield, it'll be like asking you that question about archaea, except that archaea is like a gimme. So like that one's just too easy, and then this one would just be outrageously hard, right? And if everybody misses a question, they'll just delete that question. It's not really a curve, they'll just take it out, right? So nobody gets docked any points for that. So just understand that. Like think about also what your classmates are studying because if your classmate is really going to get bogged down about signal peptidase, and all of your classmates are getting bogged down by that little detail, it's probably important for you to understand it, because just in case they give you some low yield question like that, but since everybody's studying it, then like everybody else will get it right, you'll get it wrong, and then when they review the actual questions, you got it wrong and everybody else got it right, so they're not going to take it out, right? So just kind of understand that it's important to know a lot of things. And if you have time, I would know literally everything that I wrote here, or just like everything that you write down, or everything from the lecture. But truth be told, no one's gonna have enough time to know every single word, to memorize every single detail that they learn in medical school, right? It's about understanding the big picture and memorizing things that are important because they're important to saving the lives of your patients. Okay, so thank you for listening. Thank you for watching this video. I hope that you learned something, and I hope you learned something on how to approach term one. Please reach out if you have any questions whatsoever. And yeah, have a great day.